Question 3. Nucleic acids include DNA and RNA. Each single strand of a DNA molecule is synthesized from mononucleotides. Draw a diagram to show two mononucleotides joined together in a single strand of DNA and use the symbols shown for each component in your diagram. So we have a phosphate, we have a pentose sugar, we have a base, and then we have a bond. So we know that nucleotides are assembled as a phosphate connected to a sugar, which is connected to a nitrogen base. And so we have one nucleotide and the next nucleotide. And here we have a bond connecting the two mononucleotides together. Moving on. Here they say the diagram represents part of our DNA molecule. The diameter of the molecule is 2 nanometers, and here we have the pitch of DNA, which is 3.4 nanometers. The pitch is the length of one complete turn in the double helix. There are 10 base pairs in one pitch. They want you to calculate the distance between one base and the next base on one strand of DNA, and they want you to give your answer to an appropriate number of significant figures. When we look at this structure of DNA, there are 9 gaps. We have 1, 2, 3, 4, 5, 6, 7, 8, 9 in between the complementary base pairs that are aligned between the two strands. So 3.4 divided by 9, I got 0.3777. And to an appropriate number of significant figures, my answer is 0.38 nanometers. I used two significant figures because here I was given two significant figures as well. Moving on. The diagram shows the structure of thymine and 4 other bases. We have P, Q, R, and S. Base P and S can form 3 hydrogen bonds each, and base Q and R can form 2 hydrogen bonds each. So here we're given thymine and the other bases. They say explain which of the 4 bases, P, Q, R, or S, is adenine. Adenine could be R or Q because they form 2 hydrogen bonds. However, it could also be R or S because the double ring indicates purine bases. We can see here the double rings could indicate a purine. They could be R or S as well. Moving on. Compare and contrast the structure of messenger RNA with the structure of transfer RNA. Whenever a question asks you to compare and contrast, you need to write about the similarities and the differences. So for the similarities, both contain nucleotides and both are single-stranded. While the differences, messenger RNA is a straight chain, while transfer RNA is folded into a clover-leaf shape. Messenger RNA has codons, while transfer RNA has anticodons. And the length and size of messenger RNA is variable, while that of transfer RNA is constant. So this brings us to the end of question 3.